Hello, hi. So we are going to see the hello, hi. So let's continue our survey through the Chara algae. We have completed discussion about the general features and the asexual reproduction. Is it asexual reproduction? No, the vegetative reproduction in Chara, because we know that Chara does not show asexual reproduction, but it shows vegetative reproduction through bulbils. There are two types of bulbils, amorphous bulbils and root bulbils, then amylum starch kind of thing. So we have discussed about that. Now we are going to discuss the sexual reproduction in Chara. So in sexual reproduction, what all things we have to see? We have to see everything about the reproductive organs, how it is formed, how the fertilization happens, everything like that. So for that we have to know that the reproductive organs, or the globule and nucleus, this is the structure. This is the globule and this elongated structure is the nucleus. To be more precise looking, this one. Okay, so this is the elongated female reproductive organ or nucleus, and this is the globular male reproductive organ called globule. Okay, so this globule can also be called antheridium and this nucleus can also be called oogonium. So these particular reproductive organs will be seen in the shoot of limited growth. Okay, this is the shoot of limited growth and this is the shoot of unlimited growth. Okay, so in the shoot of limited growth, at the lower nodes, maybe here, maybe here, maybe here, this is the node. Okay, this is considered as the lower node. Here we can see the occurrence of female and male reproductive organs of the genus Chara, and this will be encircled by, okay, a wall of appendages called stipuloids. Okay, stipuloids are hairy appendages at the lower nodes of Chara in the branch of limited growth, maybe as a covering to the reproductive structures. So this is it. Then we can see that this is globule in the this is nucleus. In the nucleus we can see in the lowermost region, in the comparably like bottom region, we can see there will be the presence of egg, and this is the antheridial structure called globule. At maturity it will be releasing anthrozoids. These anthrozoids will swim in water, will enter into the nucleus through the space that is present between the coronal cells. This is the corona of the nucleus. We'll see how it is formed and all. So you can see that this is a zoomed in structure from this branch of limited growth. We'll see about all these things. Now you can see that this is the branch of limited growth. In the bottommost part of the nodes, okay, this is the bottom node, this is one bottom node, this is one bottom node. In the top nodes we are not seeing that. In the bottom nodes we can see one globule and one nucleus per each nodes. Okay, this is the thing. Now we'll see the sexual reproduction in Chara. The mode of reproduction in Chara is oogamous. That is, there is a proper male gamete and proper female gamete, and that male gamete or antheridia or anthrozoid will be coming and fusing with the egg which is present within the oogonium. This is known as oogamous mode of reproduction. The male reproductive organ is globule, female is nucleus, female organ is nucleus. Then it will be having a covering of stipuloids or lateral appendages. We have discussed all these things. And one more thing, Chara can be either homothallic or heterothallic. That means maximum times these both globule and nucleus will be lying this side by side in the same thallus, but in some very rare cases it will be seen in different like thalli as well. For example, Chara vallichi, okay, Wallichi, Chara vallichi. In that, we can see heterothallic occurrence of male and female reproductive structures. Now, this is the slide that we will be discussing. Okay, if you are taking a section on the nucleus and globule, we can see that there are some parts within. Okay, there is one stalk. We have taken one section. We have cut the particular globule, and we can see this. Okay, there are some parts called shield cells. Then there is a longitudinal cylindrical rod type structure. Then below that there is some spherical thing. From that spherical thing there are some filaments coming. Okay, we will see how this structure is getting formed and how the nucleus is getting these coronal cells and these kind of cells surrounding it. We'll see everything in the upcoming slides. So a mature globule is yellow to red in color. You can see it's a completely mature thing. So it is having almost reddish color and spherical in shape. First you will be seeing globule. After completing the discussion of globule, we will move to nucleus. Each globule consists of 3 kinds of cells. First one is the outer. You can see the outer area. Outer area. You can see. Imagine this as a football. Okay. The outer curved plates is called shield cells. These are 8 in number. Okay. We'll see how it became 8 in number. Then each shield cell gives rise to a central rod-shaped structure called manubrium. Okay. I have taken. See, when we are cutting this particular globule, we can see this internal structure, and I have taken one section out of it. Okay, one section from here. I have taken one, just like we cut a cake. I have taken one section, and this is that section. We can see the topmost shield cell. From that shield cell, we can see a longitudinal structure called the manubrium originating. Below that, attached to manubrium, we can see a spherical structure, and this spherical structure is called capitulum. This capitulum will have a small protuberance to outside. It is called secondary capitulum, and that will be carrying few filaments. Okay, there may be 2 or 3 filaments occurring from one particular secondary capitulum, and each filaments will be having multiple cells. Okay, these cells will be forming antherozoids. Okay, these are antheridial filaments, and one particular cell, if I have taken from that filament, it will be like forming antherozoids. Okay, biflagellate curved shaped antherozoids. We'll see how this is formed. Okay, that means the secondary capitulum, that means this is small projection from the primary capitulum or spherical ball. We can see secondary capitulum. From this we can see filaments. So maybe 2 to 4 number of filaments may be arising from one particular secondary capitulum, and each filaments are having approximately 25 to 250 cells. Okay, one filament is segmented into different cells. We can see maximum up to 250 cells. Each of these cells are called antheridium. This antheridium will be giving rise to biflagellate, uninucleate, and coiled antherozoids. Only one antherozoid will be released from one particular antheridial filament. Now, how this kind of structure, this kind of complex structure is arising, we are going to see. First of all, at the node of the branch or shoot of limited growth, we can see one cell called globule initial. Now it will be dividing into 2 transversely. So top cell and a basal cell. Now just imagine that. Just now consider this top cell only, because the bottom cell is going to just form the stalk or the holdfast kind of structure. Just stalk. Okay, just imagine it as a stalk or basal cell. The topmost structure, consider it as an apple. Just imagine if you want 4 pieces of apple, how we will cut. We will cut it once like this, then we will cut it like in this fashion. Okay, in this way. Okay, so 2 longitudinal division, one like this, then like this. So we will be getting 4 pieces. Then we will cut it from here to get 8 pieces. So it became an octant. Okay, so this is the top 4 pieces and this is the bottom 4 pieces. So below that we can see the apical cell. Now we will see what is the transformation that is happening to these topmost 4 cells and bottommost 4 cells. Now these topmost and bottommost 4 cells, each of these cell will undergo periclinal division. Okay, it will form one division like this. Okay, it divided once like this, all cell divided once like this, then again one more division. So 2 times it will undergo periclinal division, and you can see the transformation. Like one cell became 3. Similarly, 4 such cells are there in the topmost quadrant, and in the bottom layer also we can see such a structure. Now each of the cells, okay, now we have 8 cells, right? Topmost 4 cells got divided into 3. 8 sets of 3 cells. Okay, 8 sets of 3 cells. This is 4. This is the topmost 4 cell. It is having like 3 cells each and 4 such cells. That means one cell is divided into 3. Such 4 cells are there. So in bottom also we are having such 4 cells which are divided into 3. So we have 8 sets of 3 cells. Each will go through differentiation. What kind of differentiation? The topmost cell, I am taking only the topmost cell. Okay, just imagine one cell only I am taking. This one quadrant, one of these quadrant I am taking. Now you can see that one pi I have taken. The top cell is expanded and became thick walled. This is the top cell, it transformed like this. Now the middle cell elongated and formed a structure like this, like a cylindrical rod. You can see this structure here as well. This is the topmost cell. Now this is the middle cell, it's elongated, and this is the lowermost cell attached to the middle cell. This is called capitulum. This is called this red color structure is called capitulum. It is forming one protuberance called a secondary capitulum, from which you can see multiple filaments. Okay, you can see multiple filaments, and filaments are divided into cells, and each cell can form an androzoid. So you can see this is one such a quadrant. Okay, one such a pie we have taken. Similarly, we are having 4 cells, 4 such divisions in the topmost quadrant, and like in the topmost quadrant, and even in the bottommost quadrant we are having such 4 cells with 3 cells of this kind of transformation. Now everything will aggregate to form a structure called globule. It will form a structure called globule. Okay, when everything is coming together, this is one cell. It was divided into 1, divided into 2. Here it was also divided into 1, divided into 2, and it each formed structure like this. Then everything aggregated together to form a hollow, like a football like shape like this. So this is the thing we have to draw in exam. We need not draw like this. We can like draw only such a section only. Okay, then we can write that. Imagine 8 such differentiated structures coming together. A ball-shaped structure which is known as globule is formed. Each cell will be forming an antheridial filament which will be producing an androzoid or sperm. So this is about globule. Okay, now we will see. This is one more explanation, one pictorial explanation of the same thing that I have explained. You can pause the video and read this. One more point: each of the filament or each of these filaments will be having multiple cells, I told you. So each will be producing one androzoid each. So from one complete globule, we can estimate to get around 20,000 to 50,000 androzoids. What a huge number, right? Now coming to, this is the globule and nucleus. We have seen this figure already. Now coming to the nucleus development. Before that, you can see that in what all ways people represent this thing. This is one mode of representation of this globule, but we need not draw a complete sphere. We can have one cell's expansion like this. Like this only you can draw one section. You need not draw such complex figures. Now coming to nucleus structure. So nucleus is the female sex organ. It is having an elongated oval shape that is having a small stalk, etc. We will see how nucleus is formed. Similarly, like the globule, there will be one initial cell. It will be dividing into 2 transverse division. The basal cell will be acting like a stalk. It will not be dividing further. The topmost cell will be having one division and with a large egg cell and a small supporting cell. So let it be like that. The middle cell will undergo 5 tangential division. Imagine that you are biting an apple from 5 sides. Okay, 5 tangential division, and it will be from each tangential division one filament will be formed. That filament will be like this, one tube like section and one segment here. At the tip there is one more cell. Okay, one more cell. Now each of the filament will be forming, the elongated cell will be the tube cell and the tip segment is known as coronal cell. Now all these middle cell filaments will be encircling the egg. Okay, encircling the egg, and the tipmost corona, the topmost corona, it will be coming to the top like this, like a head, like this. Okay, so the remaining tube cells will be encircling the egg. Okay, so that's why this structure is formed. Now during the fertilization, what will happen? This particular globule will release the androzoids. It will swim and it will be like entering inside to fertilize with this egg through the gap that is present between the corona. So this is about nuclear development. Now the zygote is formed. This is a zygote that is formed. We will see a more clear figure in the upcoming slide. The zygote will be 2n nuclei. It will undergo meiosis to form 4 haploid cells. When the 4 haploid structure is formed, there will be one division like this, and 3 will be on one segment and 1 will be on the other segment. These 3 will be degenerating, and the existing or surviving one cell will be dividing into 2 nuclei, and 2 cells will be formed. One is called the protonomal initial, which will be forming the new thalli. Then the second will be the rhizoid initial. It will be forming the rhizoid. So it will complete the formation of new thallus. So this is about